because I said what was on my mind. I made a lot of enemies. I made a lot
Welcome, everyone, to Science from the Fringe. My name is Bryce Nickels. I'm a professor of genetics at Rutgers University, and I'm thrilled to be joined today by Kevin McKernan. Kevin is a biotech entrepreneur and inventor. He is the founder and chief scientific officer of Medicinal Genomics Corporation, which published the first genome of cannabis back in 2011. Kevin, welcome to the show. Can you tell me a little bit about what you were doing before you founded Medicinal Genomics Corporation? I will say you also have a degree in biology from Emory. Is that correct?
That's right. Yes. Yeah. I dropped out of some PhD programs along the way to start some companies and never really got back to the academic path, unfortunately. But I wasn't so fringe in 1996 is when I started at the Whitehead Institute, the Center for Genome Research there. I was the team leader for research and development. So if you remember that big race with Craig Venter and the public sector, I was building the robotics pipeline and the plasmid purification systems and sequencing chemistry for Whitehead. A lot of robotics and engineering, which was fun. But I guess I don't think I really went fringe until medicinal genomics. There's a few years before that, from 2000 to 2006, I started Agencourt Biosciences Corporation. That was we were DNA sequencing, like a contract sequencing facility, and we also made all these DNA purification kits, these magnetic bead kits you might be familiar with, Ampure and Spry, these tools we sold to the marketplace. And Beckman Coulter came and bought that company. But we had a little skunks work project to build a DNA sequencer they didn't know how to value. And so, because it was kind of a shoot the moon project that turned into the solid sequencer. So we spun that out into another company called Agencourt Personal Genomics, and 19 of us moved across the hall and built that platform and got it to sequence E. coli and presented that at AGBT. And then Illumina and ABI got into a bidding war over that company. And ABI prevailed, and I ended up going to ABI for 5 years, where we brought the solid sequencer to market and played around with Ion Torrent as well. We acquired that platform towards the end of that. I stayed for about a year during that integration, and then that's when I went fringe. I guess I went off and sequenced the cannabis genome, and that's where I am now.
Well, now, to be fair, it's not. I don't only talk to people that are considered fringe, and I don't necessarily think that you are necessarily fringe. In fact, that's one of the reasons I wanted to talk to you, because you have been branded, I guess, as a fringe molecular biologist, or I don't even know what but they've called you all kinds of things. And
Yeah,
I think
sure.
that's really what I wanted to talk to you about.
Fringe is a badge of honor now, right? It decorates the head of the NIH. So,
That's true. Although I believe, well, actually, I think the NIH, the building where the NIH director's office is, I think it's Center Avenue or Center Street. So it's actually, he's gone from being the fringe to being the center.
oh, that's funny.
Which is, yeah, interesting. So I still remain on the fringe, and that's why I'm here with you, another possible member of the fringe. But I think the key thing is that you have decades of experience, like with molecular biology, with sequencing, with building companies, with running companies, with developing technologies. You're not like, well, I don't want to say you're not some crazy person or whatever. Like, you have bona fide chops as a scientist and as a reputable scientist. And yet we sit here today with you as the target of a lot of vitriol, and I don't even know what to say. It seems like coordinated efforts to discredit you because of some of the recent work you've done looking at the presence of DNA contamination in the mRNA vaccines. Do you want to talk a little bit about that?
Sure. It actually started at the beginning of COVID. When the first PCR paper came out from the Corman Drosten paper, I was a bit appalled by it because we're at Medicinal Genomics. We make PCR kits for the cannabis field, and we'd never get away with what they did. I mean, there was no internal controls. All right, so in PCR, you kind of always have to have a target you know is going to be there to quantitate an unknown. In the case of PCR for diagnostics in humans, you usually target the RNA-P gene in humans. It's a housekeeping gene, and you measure the viral CT compared to that. And without that, you don't really know how much virus you're scraping out of someone's nose. Those swabs can vary 10,000-fold on just the RNA-P gene alone. So that means the swabbing of the cells in your nose is really variable. And for those not familiar with tenfold. Actually, it's more like 1,000-fold, which is about 10 CTs. So you can have a 10 CT swing in how well the swab behaves. So if you set a CT score at like 35 to call people positive, you're really 35 plus or minus 10 CTs,
What,
which
Kevin, what's the CT?
varies.
Can you just refresh people's, like, what is a
Yeah,
CT?
that's the cycle threshold in PCR. And so when you get out past 35, you're getting close to like below 10 copies of a target, sometimes a single copy, depending on the assay. And so you're getting really close to the noise floor of the assay. And we now know, and we suspected this back then, that a positive CT score, let's say below 35, didn't mean you're infectious. It could meant the virus that it was, you had it a couple months ago. The viral, you're only infectious for like 9 days, and you're PCR positive. You can be PCR positive for almost 90 days. So if you're not careful with PCR, you're going to flag everyone who's already immune and quarantine them, and then you're going to destroy herd immunity as opposed to taking people who are infectious out of the community. So we spoke up about that, being like the test needs to have an internal control. There's some mistakes in the primers, and there's some false positives that were showing up in a lot of papers that the water samples were testing positive. So, but that created a huge scare at the initial pandemic, that it was bigger than it really was, which resonates with what Jay was finding. When he went and looked at this from an antibody perspective, he was like, "Wait a minute, we already have like 2% of the population that's positive. That means this thing's already come through here, and that means that the IFR that people were assessing from the Diamond Princess was meant that this is not nearly as deadly as anyone anticipated, perhaps tenfold less so." So we got attacked for doing that. We wrote a sort of a Corman Drosten critique, and that paper got just torn apart, and it created a lot of enemies online.
Did you publish that paper, or was it just like a
It
note?
ended up on like a preprint server, and it got complicated getting it through peer review. We tried at one or two places, and the person they had come in to review this had their own conflicts. So that the whole story around that initial Corman Drosten paper was somewhat conflicted, and that the people who wrote the paper were on the editorial board of the journal and had PCR companies in the wing waiting to do PCR and commercialize this. They got through a 24-hour peer review on the journal that they're the editors of, and they didn't disclose those conflicts. So we were kind of raising some alarm about that. And then I think Stephen Buston weighed in to shoot it down, although the arguments he used to shoot down our critiques were the same arguments that he levied against the same problems they had with the. He was also a person on the court case against what is his name on the autism side that got skewered, Wakefield. Andrew Wakefield. So he was paid by GSK as an expert witness in the Wakefield trial, and he tortured Wakefield for all the same problems that we were raising against Drosten, but he had a totally different opinion on Drosten when we levied the same critiques that he used against Wakefield. So it was a very bizarre review that occurred. So it never really made its way into a final publication, because some of the authors went into the no virus world and suddenly couldn't sign their authenticity documents. When you submit a paper, all the authors have to agree on its authenticity, and a few of the authors that were on there were like, "No, I no longer believe viruses exist," so they couldn't sign it.
I guess just to back up for a second, you entered right after COVID hit. Like, you took a look at the use of PCR tests as a measure of the extent of, COVID infections in the population, and said, wait a second, these measurements, the way in which you're detecting COVID might not give an accurate, might not accurately reflect what's truly going on in the population.
Yeah, it's going to overcall it.
Right. And that's because PCR, you can use that as a tool to detect a single molecule of a nucleic acid.
And you can't discern live versus dead. I mean, there are methods that exist and have been published for decades that can discern things that are in capsids or not in capsids if you prepare the sample correctly, but they weren't using those.
But but what you pointed out, which made me want to go vomit during this interview, which would be unfortunate, is the conflicts of interest that people had for being able to use PCR as a means of detecting the extent of, people being, ill with COVID. And if they had massive financial incentives to be able to use that test, then you're questioning whether that test is valid is going to present a problem for them.
Yes.
Um,
Yeah.
and that seems to be a recurring theme, I think, with what you've done more recently with the DNA contamination. But going back this PCR thing, isn't that there's a very, there's this clip of Kary Mullis talking about PCR that circulated on Twitter all over the place. Can you put that in context?
Yeah, I think where people perhaps take a little bit of liberty with it, because if you look at Kary Mullis's patent estate actually shows that he believed in PCR for as a clinical tool. But I think what he's stating there is if you overrun the assay, it'll pick up. Like if you go up 40 or 50 cycles, you'll always get a signal. And you can't really discern the health of the patient from that. It might be a confirmatory piece of information next to the fact that they have symptoms or antibodies or something else. But in isolation, when you use this on asymptomatic people, you're going to overcall the population, over quarantine the population. So parts of his video that circulated were true, and that you can't run this thing out in an infinite number of cycles unless you really validate that you don't get false positives out at 50 cycles. And the bit of information you get from there should never be used to quarantine an asymptomatic individual, because you just don't know if they've recovered or whether it's the front of the back end of the infectious curve. So I think it got abused in the pandemic, and that they started applying this, like, CVS's and in parking lots where there aren't any physicians involved, to ask, wait a minute, is this person even symptomatic? And so they start flagging people, and then there became a financial incentive for high positivity because they put in contact tracing. So every person you had a positive score from, you'd get 3 or 4 more tests from their family. So no one wanted to move the CT scores back to what they would sequence at. You'll notice the CDC wouldn't even bother to sequence a sample unless it was under a CT of 30, because they couldn't. There wasn't enough DNA there. So that meant all these people that were getting called after, I think they even backed it down to CT of like 20. So everyone being called after a CT of 20, they had no way to sequence confirm that those were actually real hits. They were just PCR signals at that point. And that ends up being the large majority of cases that are declared a positive COVID test. And I think this weighed into a lot of the sort of a snowballing effect in the hospitals, because they would find all these people that are COVID positive and start flagging them as being COVID when they're really dying of all these other comorbidities, and they happen to have some COVID RNA floating around in that environment, that they'd flag them as COVID, that they get more reimbursement if they were flagged as COVID. There's just a whole cascading event of things that would occur that probably wrecks the healthcare system by misappropriating ventilators and remdesivir and all these other morbid outcome procedures.
So that is what I guess could be referred to as PCR-gate, right? So you
Yes,
were
yeah.
involved in PCR-gate, and then you learned your lesson and you just went away. You said, "You know what? This is too difficult to deal with this criticism. I'm just going to leave COVID altogether." But no, that would have been the smart thing perhaps, but that's not what you did. So so that you didn't publish this paper, the people that you were co-authors sort of decided that viruses don't exist perhaps a little, but some
Yeah, a
of them.
couple of them. It only took a couple of them, and we'd have to, like, strip the authorship apart and rewrite the whole thing, so it just got stalled at that point.
And then when did you pivot into the DNA contamination stuff?
So that was somewhat accidental in that I after that, I went and published a paper with Peter McCullough on the differences between the virus's spike sequence and the vaccines. They codon optimized the vaccine, so they were actually very different RNAs. The RNAs end up making a lot of hairpins, and that's by design. The actual codon optimization tends to make more rod-shaped RNAs so that they survive longer. And there's some artifacts when you make rod-shaped RNAs like that. Not
Well,
only
so
is it
just to be clear, what you're talking about, they're forming hairpins, or these are also like secondary structures.
yes,
RNA is normally a single strand, and then if there can be pairing between, like, I guess, is that like intramolecular pairing?
yes.
Or actually intermolecular pairing too. That, and in that paired form, it's less susceptible to proteins, enzymes that would chew it up.
That's
in the
right.
cell.
That's right. And so it was more resistant to RNase-L. It created a lot more G4s, which are quadruplex G structures in the RNA that has different, biological consequences, and it probably has a completely different.
Wait a second. G quadruplex, has that been shown to actually happen in cells, or is that just in vitro?
No, this is just that's just from modeling. Doing it in cells
Right.
is kind of tricky.
I don't know if anyone's ever shown a G quadruplex is formed in cells. At least but several years ago when I was interested in this as it relates to transcription. I don't know if anyone.
You
Did
need, yeah, there's a certain particular procedure that you need to do to measure those things, and it's
okay.
non-trivial. But you can,
Everything's hard. Yeah, this is the thing. Actually, my career, most of my major findings have been just showing in my career have been showing that something that's been observed in vitro actually happens in a cell. So that's kind of like one of my
oh yeah,
little.
yeah, yeah. Well, it's good
Yeah,
for you. That's the hard part.
it's the hard part. Yeah, exactly. So anyway,
um,
sorry, go on. You mentioned G quadruplexes and it triggered me. So
yeah, yeah. So those can create different. They can create problems for the ribosomes. All right. So when they fold like that, the ribosomes don't like to translate very efficiently down there, and what has been shown in cells now through the Mulroney paper and a few others. Postnikova showed this as well. That when you code and optimize things like this, you get ribosomal stalling and you get frame shifting. And the frame shifting is also exacerbated by the fact that they put in this novel nucleotide into the RNA, this N1 methylpseudouridine, and so that changes the TM of the RNA, and it makes it fold back in itself a lot more. It makes the T7 polymerase end up doing a lot more template switching and loop back synthesis. And so the Mulroney paper showed that frame shifting is actually happening. And that we predicted that in that paper. Again, a preprint that went through peer review had two reviewers sign off on it at a Hindawi journal, and then an editor at the last minute killed it. Uh,
but by the way, all of this stuff isn't that, this is all basic, molecular biology, basic
yeah,
biochemistry. There's nothing like controversial. You're just characterizing how nucleic acid structure affects the ability of the protein
yes.
translation. None of this should be that complicated. It's like this isn't out of left field or anything. This is
No,
stuff
I
that.
was using standard tools like RNA fold. RNA fold's been used in our field for decades, and showing that, hey, look, these things fold differently, and they're going to create different structures that the whole microRNA machinery is gonna view differently. And so that needs to be considered. Its biodistribution is going to be broader because the viruses stick to certain cells and the LNPs go everywhere. And the frame shifting has been documented to happen with this modified nucleotide. I zeroed in the modified nucleotides because I've built sequencers, and I know from experience, so if you put a single methyl group on a base, everything goes out the window. Like enzymes all treat that differently. They're very sensitive to a change like that. So
Well,
I
in
knew.
fact, it's the reason why they use that is so that it doesn't get chewed up in the cell,
Right.
like it's more stable.
Yeah, yeah. So that's affecting RNase-L, right? So that enzyme doesn't shoot as well. But if it's affecting that enzyme, it's probably going to affect the ribosomes as well that have to read this stuff. And there's papers out there from Parr et al. Colin Parr showed that if you just put 4 of these pseudouridines inside of a 25-mer oligo, the TM goes up like 9C, which is a huge shift in the melting temperatures. So that means it's going to be much stickier and binding to itself more than traditional RNA. So we wrote about all these issues in that paper. It got through peer review until an editor decided to kill it, and
But on what basis? That it wasn't
they didn't
important
give us
enough,
one.
or
They didn't give us one. The reviewers signed on, and the editor said this needs to go through an additional editorial review. Now it turned out we learned like a few months after that Hindawi was in the process of retracting 600 papers. And so it could have been that their journal was like under some type of heavy scrutiny, and they just didn't want another controversy going out the door during this. So I suspect that probably weighed on them. But they never gave us a solid answer as to why they were giving this an additional round of scrutiny. So
but
um,
wait, Kevin, real quickly, I realize I didn't ask a basic question here. How are you supporting this research that you're doing? Like, how do you fund it? This is all self-funded, or?
this is, yeah. This is all in silico work, so I could do this on the weekends and at
Oh,
night.
so you made. I see. So okay. So most of the things that you're putting out are based on in silico.
Yeah, until we got to the vaccines stuff, that's when it turned into actually some wetware, and yeah, that I've just been funding myself. But And we've taken a fairly low budget approach, as you can tell from the work that we put out. We don't have expensive mass specs here, and I can't afford to go buy one. But after that McCullough paper came out, people started sending me vaccine vials anonymously in the mail, saying, "Hey, look at these." And I had no use for them, so I just threw them in our freezer. And then maybe 6 months later, um, we had this project where we were doing RNA sequencing of cannabis plants out during an infectious cycle to see how hop latent viroid was destroying the growth of these plants. And we got some reads back where they didn't look like they're coming from RNA. We're getting sequence that was all over the genome. And that's usually a sign that you screwed up your RNA prep somewhere. So go back and spike in a poly-A RNA that's a control, that's pure, and see where in the pipeline that thing doesn't come through. And so I'm like, all right, I need to go get some poly-A RNAs, and I'll spike it into my RNA-seq assay and see what's broken. So I had those in the freezer, and I'm like, "That perfect. I don't need to buy them. I'll just hit these with Triton X and throw the RNA into the RNA-seq, and when they come out the other side, it'll tell us whether our mag beads are broken or our DNase isn't working." And it turned out the DNase wasn't working. Ironically, that's going to be a theme you'll understand when we get Pfizer stuff. So when we get those reads back, and we look for that RNA, and the only reference genome there was from Andy Fire. His group had put out the sequence of the vaccines, but they didn't publish the reads. They just put forward a FASTA file of what they thought the vaccine was. And so we mapped against that and said, okay, it's there, but our IT guys in bioinformatics were like, you can't just map, you gotta assemble the whole thing. So they assembled all the data, and that's when the plasmid popped out. And we're like, oh, wait a minute, there's a whole plasmid backbone in here and there's parts of this plasmid backbone that nobody knows about. It's not documented. It's like the whole SV40 area of this plasmid was not in the EMA documentation for the plasmid. So then
What is SV40 again? Like, what is that? That's
so SV40 is a component that was taken out of the SV40 virus. The SV40 virus is about 5,000 bases long. We only have about maybe 400 bases of it inside this plasmid, but they took some interesting components. They took the promoter, the enhancer, the origin of replication, and the poly-A signal out of that virus, and put them into this plasmid to help them drive the kanamycin resistance. So, when you take a plasmid and you put your spike region into your plasmid and you want bacteria to replicate it, you have to have a kanamycin resistance gene to give that E. coli resistance so it will copy your plasmid. And they used SV40 as a promoter for that kanamycin cassette. It didn't need to be there. Moderna didn't use that. They used a bacterial promoter known as AMP-R, and that's a safer thing to do. But what we learned by combing through the literature is that Pfizer, in their race to get this to market, called up their gene therapy department and got one of their plasmids. And these SV40 plasmids, that's what they're used for, is they have a nuclear targeting sequence, and they're known to integrate into the genome. And so they grabbed one of those.
But just to be clear here, all they really need, like you just said, which is this is where like just don't, what is the quote, like don't attribute like something that's maliciousness when it could just be incompetence. But
Yes.
so in order for, so like the plasmid that they're preparing is they're using E. coli or bacterial cells to prepare a lot of these circular molecules, these plasmids. And in order for that plasmid to be or for the company to be able to say, okay, the bacterial cell has the plasmid carries a resistance gene for kanamycin. Cells that contain the plasmid are resistant to kanamycin, and the kanamycin resistance gene is driven by these sequences, these SV40 sequences that they didn't have to use because they were just lazy. And it's not a conventional thing to use because, like, I'm a bacterial geneticist, and I've never encountered use of SV40 as something to express a antibiotic resistance gene in bacteria. So I didn't realize that little nugget of information. That's pretty disappointing.
Yeah. Now, if you're really into plasmids, I spent my career at Whitehead dealing with plasmids. They do have an AMP-R region in their plasmid as well. It's just like another 100 bases back, and I don't think it's going to be close enough to the kanamycin gene to be effective. Like with bacterial transcription, In translation, there are really important constraints on where the promoter is to the gene. It has to be like -35, and it has to have a certain like Tata box and these Scheindelk RNA sequences. So from the way the plasmid is laid out, it's a shuttle vector that's designed to operate inside mammalian cells and inside of E. coli, and it can operate either kanamycin or neomycin. So something
I
that's
see. So they wanted expression in mammalian, so I see. So
yes.
that, but they didn't. Okay, so I'm
Yeah,
confused
you
if
don't
they
want
ever.
that in a vaccine, but I think they just didn't have the time or care to delete those regions when they realized this was going to go into E. coli manufacturing and eventually end up in a mammalian host.
By the way, once you get once you select a plasmid containing bacteria that for antibiotic resistance, you know how hard it is for it to actually lose it, even when you remove the selected pressure. Have you noticed that? I don't even think you
Yeah, the
need
copy
once you
number, it doesn't go away. The copy number might go down, right?
possibly right, but it's yeah. So anyway, like I mean, they didn't
This thing also has two other origins of replication in it, so it's not going anywhere. It's got a coli-1
right.
origin, so that'll replicate in bacteria, and an F1 origin that'll make single-stranded DNA for phage.
But okay, but the other thing, okay, maybe, and I know we're bouncing around a little bit and we're wonking out with the science here, but I was going to say one of the things when I heard about your work initially and how you were characterizing DNA in the vaccines, um, and that it's plasmid DNA,
Yeah.
but that was unexpected in part because there was a difference in how the RNA vaccines were manufactured for the trials
Yes.
versus when they scaled it up. So to me, that was when I learned that, and as somebody that like is very familiar with using linear template or supercoiled template, I was astonished that that had happened. Do you want to get into that a little bit?
Yes. Yeah. So for the trial, what they did is they amplified the spike region with PCR, and that just amplifies like 4,200 bases. And as you know from your experience with this, when you amplify something, you raise that signal above the background plasmid, maybe a millionfold. So the background plasmid gets drowned out during the amplification process. But you can also anchor like a biotin on the end of the primer so that the DNA template is easily removed from solution when you're done. Right? You just use a magnetic bead to scrape it out. So they had a very, very clean process called Process One for their clinical trial. Then when they went to scale this up, they didn't want to do the PCR step, so they skipped it, and they just went with purifying the plasmid right out of E. coli. Now, that comes with a whole host of other problems. One, the entire plasmid is there, and as you know, T7 polymerase is a linear reaction, so you have to feed a lot of DNA into a T7 reaction in order, and particularly if it's supercoiled, it's harder. But that also comes with endotoxin, right? When you crack open E. coli cells, your plasmids are very rarely free of endotoxin. And endotoxin can create anaphylaxis on injection. And that's what we saw with this when this trial scaled up and they started going to the public. People were dropping in the with anaphylactic shock in the vaccine centers. So the
And you
end,
think it's because of the difference in the I mean, sorry, your speculation then at the
yeah.
time was that it was has that been shown or the people
No,
just
so the challenge with the endotoxin measurements is they're all redacted. All the documents we've tried to get from the EMA, they've redacted those numbers. And I've tried to measure them with some LAL assays, but it's hard to do it when there's LNPs around. The LNPs, I think, coat the endotoxin. I think ends up inside the LNP, and so the LAL assays don't operate. They trigger a lot of false positives. So I
But
don't.
Kevin, what happened? How does this allowed to have something I feel like this is something that's shameful that it has the fact that they did a trial with one thing and they report the results of that trial and then they give to the public something that wasn't
Totally
actually
different.
tested in the trial.
Yeah, I don't know how they convinced. The EMA was aware of the change. The EMA is the FDA version of in Europe, European Medicines Agency, and they asked them to do another study of like 250 people, and Pfizer never reported those numbers to the EMA. Now, first off, 250 people is not enough to find one adverse event in 250 people, right? It's statistically really not. You won't find anything in a study that small. But the fact that they didn't report anything back to the EMA makes me think they did find 1 or 2 adverse events in 250 people and didn't want to show them the data. And by then, the vaccine campaign was marching along, and they were using the excuse that we've already given this to millions of people, so what we can't stop now.
Right. That's to me. This also is this idea that like we have to like segment the population into pro-vaccine, anti-vaccine. This piece of it shouldn't be controversial in the sense of if you've changed the way in which this, drug or whatever vaccine's been that's been given to billions of people was manufactured versus a trial, like that shouldn't be a controversial thing for the public to have questions about it and demand answers about what truly is the safety of that relative to what was tested initially. That really kind of, I mean, to be frank, it really pissed me off when I just heard you initially talking about that and how why should that be controversial that the public should have concerns about that? But
I tend to hide behind, oh, we told the regulators about it, so it's on the regulators. They knew
that's
that
fine. Well, then the regulators, right? So then, fine, pass the buck in terms of accountability. But this is if you're saying that the public still doesn't know what the difference is, like what effect that actually had on the safety of the product.
it is, and I don't know why the regulators are still backing this because they do seem to be passing the buck to them and throwing them under the bus. But at the same time, another thing that became very apparent to me in their the way that they operated with the regulators is they hid things from them. So this whole SV40 region was not on their plasmid map. And if anyone who uses these tools that annotate plasmid maps, if you plug this sequence into Snapgene, the first thing it's going to annotate is SV40. So somebody at Pfizer had to go in and remove it to defraud the FDA. And they gave them a plasmid map that didn't have SV40 in it, and then handed them a sequence that had it in it. And the FDA just never threw that sequence into Snapgene to see if everything on the map checked out.
But couldn't it have been just incompetence rather than deliberate malfeasance? This kind of gets into maybe I can see where people like if you're implying that there was malicious intent, that might get you in trouble in a sense of where they come after you. But if you're just pointing out that there's this abject, what appears to just simple incompetence, wouldn't that I don't understand why everybody doesn't support this, like support
I think
you and your efforts.
I think there's a good case that it's intentional deception, just because in that plasmid map they have the kanamycin gene annotated and some other components. And these software tools don't selectively annotate different regions of maps; they just annotate the whole damn thing. And so someone had to go and erase it before giving it to the FDA.
Have you
And
spoken with people at the like, so, I mean, Marty McCary, I assume, would be involved, trying to
I haven't spoken to him. No,
okay.
if you've got an introduction, I'd love it. I know Tracy who's there, and
Oh,
she was
right, yeah, Tracy's there. Have you
um,
spoken with her? She must be aware of this stuff.
I spoke to her before she got into the FDA, and she was quite shocked by it. And now she's in there and claims to be taking it a lot more seriously. So hopefully we'll see some change. That they'll realize that look, you got Pfizer hid stuff from you, and that means you can't sit there and trust their PCR numbers, which is what they have been. That's been their position to date, which is that the numbers for the DNA contamination are under the limit because Pfizer said so, not because we measured them ourselves. But there were kids, some high school students, that went into the White Oak facility in the FDA and wrote up a really interesting paper in the Journal of High School Science saying they found the DNA contamination that was over the limit in the FDA labs. So there's now this debate gets a little bit nuanced as to how you measure the DNA. You get different numbers depending on how you measure it. All right. So they're contesting the use of fluorometry to measure the DNA, despite the fact that they use fluorometry to measure the RNA. The dose that's calculated in all these shots is done from ribogreen off a fluorometer. But if you turn around and use picogreen to measure the DNA, they call foul, saying you have to use PCR. And that doesn't line up with Moderna's patents. Moderna, from Stephen Bansel's on a Moderna patent saying you can't use PCR. It won't measure the whole plasmid. It just measures one region. And these nucleases they use, like DNA Swan, doesn't chew the plasmid uniformly. It has
Oh,
hot
right. So go back to that because I think I cut you off before you were getting to this. You were pointing out that when they initially prepared the RNA for the trials, they did it in a way in which it was easy to fish out the DNA that was used as a template because
Yes.
they had biotin or something on the oligo that was used, so you could use streptavidin beads or whatever to fish out and get rid of it. But when you now use a plasmid, what do they do to try to get rid of the plasmid DNA from separate it out from the
So
RNA?
they were throwing in the wrong enzyme to get rid of it. They threw in DNase-1. And DNase-1 is a nuclease that chews DNA, but it cannot resolve RNA-DNA hybrids. There's a paper from Sutton et al. That explains this. So you should anticipate RNA-DNA hybrids because you just made a lot of RNA off of the plasmid, right? You did a T7 polymerase reaction. You've made 30 micrograms of RNA off of maybe a microgram or two of plasmid. If you go to hit that with DNase-1, it's going to chew the backbone of the plasmid that doesn't have any RNA, but it's not going to chew the spike region very well. And so if you measure with qPCR a region outside of the spike region, you'll get a low number. But if you measure inside the spike region, you'll get 100-fold more because the nuclease isn't working in that region. So that's the problem with using PCR is it only measures 100 base pair region, and then they extrapolate that, okay, it's a 7,800 base pair plasmid, multiply the number by 78, and that should be your total nanograms. But that's just misguided because the DNases don't chew that DNA universally. They have biases in where they chew. So there's other nucleases they could have used. I mean, there's a handful of them in the NEB catalog that do a better job with DNA RNA hybrids, and they would have destroyed all that DNA with the proper enzyme.
But wouldn't it have been better, though, to not try to chew it up at all, but rather to sort of fish it out?
Yeah, absolutely. It would have been much better because this is the thing about there's a bit of a paradox here that it's good to chew the DNA up if you're going to do a naked DNA injection, right? Because the nucleases in your body will have an easier time destroying 200 base pair pieces of DNA than 7,000 bases, right? And but the moment you move to an LNP, and you protect that DNA, it's actually worse if it's fragmented because now you have more phosphates and hydroxyls that can integrate. Right, the actual ends of the DNA are what are the business end of integration. So by chopping a big piece of DNA up into many small pieces, you create a lot more phosphates and hydroxyls that can work with ligases or polymerases to integrate in the genome. So you want it really small if it's going to go in naked because your body will take care of it in 10 minutes and destroy it. If it's in an LNP, it's going to get into the cytosol, and now you have a different beast on your hands. You've got shrapnel or buckshot that's going into the genome that has a lot more reactive phosphates and hydroxyls.
Did you also maybe I saw you speaking the other day about there's like a immune response that's triggered by these
There
molecules?
is, yeah. There's one known as the CGAS STING pathway. STING stands for stimulation of interferon genes. It's like a cyclic GMP AMP STING pathway, and that Kwan et al. KWAN has shown that if you chronically hit cells with that, it's going to trigger that pathway, which can lead to oncogenesis. So it doesn't need to integrate for this to be an oncogenic risk. It just needs to get into the cytosol, which many people have now shown it does. Ulrich Kammerer has taken these vaccines and put them into cells and then put them through several cell passages and can find the SV40 signal like 3 divisions out or three cell passages out. The signal goes down over time, but it's still there and it's maintaining itself to some level after you've transfected these things. She doesn't know if it's integrated. She didn't do whole genome sequencing. She just did qPCR to measure this over several cell divisions and can see SV40 signal there. So we know it's in the cytosol. We should expect it to be there because when you package these with LNPs, they're most likely going to package with the RNA. Particularly if they're self-complementary to the RNA, they're going to be packaged as an RNA, an RNA-DNA hybrid into the LNPs.
But how many? So I guess when I hear you talk, I hear a lot of. So like you began saying you were doing a lot of computational stuff and like theoretical modeling. How much of you're generating a lot of hypotheses, correct, for how these things could cause problems in cells. But how many of these hypotheses have you been able to test?
So that's a good question. I on our end, our facility here, we can do a lot of measurements of DNA and RNA. We don't have mammalian cell culture here, so that's been happening. And Philip Buchholz has done some work on organoids, and Ulrich Kammerer has done some work in cells. So most of the confirmation of the cellular impact of this has been happening in other people's labs, not ours.
And how are they getting money? Are they being supported by private donors for that?
Yeah, I don't know how some of them have, have either grant money that they're using to teach students and use this as like a teaching project, or have private funding, I suppose. But I'm not really well versed on either of their funding sources for this. But I don't think it's very fun. It might be fundable now. It wasn't fundable when we were doing this.
Right. Well, that's why I was amazed that I mean, the incentive for you to do this was very low.
Yeah, the reason we doubled down on measuring this with, I mean, we went through and measured it with Oxford Nanopore, with Illumina sequencing, with Agilent tape stations, with a nanodrop which is a UV spec, fluorometer, qPCR. We hit it with everything we had in our laboratory because we knew if we put this out and we were wrong, it would just destroy the company. We get sued and it'd be a big mess. So we knew we couldn't hide it. Once we found the plasmid, we're like, crap. Now we're pregnant with this information, and if we scuttle it, it's going to be a disaster for us as well. So we just doubled down and tested this every which way we could in our facility, and then put out a preprint, which got flamed and attacked from every direction, as you might imagine. But quickly, people on Twitter started reproducing it. Philip being one of the first, and then Sin Lee did it as well in Connecticut in his CLIA laboratory using Sanger sequencing and completely different methods. And then slowly, we started to see Bridget Koenig came out and found it. Didier Riault found it. Ulrich Kammerer found it. And it's just a big cascade from there of all these other people finding very similar numbers. But the big debate where it rested with our paper, and the reason it took perhaps longer through peer review, is that when we first did this work, we didn't have an RNase involved. We did some RNase work with the qPCR side, but not with the fluorometry. So people were contesting, and rightfully so, that the fluorometry could be overestimating the amount of DNA that's there because those dyes could have crosstalked with the RNA. And the manufacturers do have some specs, so there's about a 1% crosstalk with RNA. So we figured it's not playing a huge role given the numbers we're seeing. But we went back and confirmed and used an enzyme that erases RNase A, and then remeasured it. And the numbers come down about an order of magnitude when you do that, but there's still an order of magnitude or 2 orders of magnitude over the FDA's 10 nanogram limit with fluorometry. And so most of the attacks in peer review were that you can't use fluorometry, but very few of these people had actually read the EMA documents that showed they're using fluorometry to measure the RNA dose. And those dyes, like ribogreen, has a much higher crosstalk with DNA than picogreen has with RNA. It's something like 50% of the signal from ribogreen can crosstalk onto DNA because these things they tend to stick to minor grooves, right? At least picogreen does, and then DNA is double stranded, so it has a minor groove. And I think where the whole field got a bit sidelined is that they expected this RNA from Pfizer to be single stranded, but as I mentioned earlier, now it's designed to hairpin, so it may attract the picogreen dye with a lot more affinity than what the manufacturer is anticipating. So you do have to erase it and measure it correctly, which we finally did, and it's now through peer review. And now we're at that stage of once your paper's through peer review, the pub smear mob comes and attacks it to try and get it retracted.
Right. So you're talking about this paper that just appeared online called Quantification of Residual Plasmid DNA and SV40 Promoter Enhancer Sequences in Pfizer/BioNTech and Moderna modRNA COVID-19 vaccines from Ontario, Canada. And you're one of 3 authors with David, is it Spike? How do you pronounce it?
Speaker. Yep.
Speaker and Jessica Rose. So,
Yes.
And this is the first peer-reviewed paper you've published on the DNA contamination?
Yes. Yeah, this went through probably 5 other journals who rejected it at the desk. They wouldn't even send it out to peer review.
And this is in autoimmunity.
Yes.
Now, before you get into this, let me this is crazy to me that this even has to be something that you're doing. Why do the vaccine companies not join you and say, hey, thanks for pointing out that we might have some issues with our product. Let us help you figure out what's going on so we can give some reassurance to the public. Like how come that doesn't happen?
So that has happened with one paper in Germany where BioNTech funded a group. The paper's called Kaiser et al. And that paper, I think, intentionally missed it. They went in and purified the DNA first with an ethanol precipitation and a phenylchloroform precipitation. And if you're familiar with those precipitations, I spent a large part of my career building DNA purification tools at both the Genome Project and at Agincourt. And when you try to purify small pieces of DNA, most of the kits out there are designed to eliminate them because they're trying to get rid of primers. And so if you're not careful, you're going to wash away all the molecules under 200 bases and say, voila, there's nothing here.
And literally everybody that does molecular biology, gets trained in molecular biology, knows this.
This is the reason we do it, right?
Yeah.
It's like
This
you
is
got to get rid of
not-
your primers. You got to run
Right.
it through a column that gets rid of the
Right.
primers.
So
Well,
all
that's-
of the scientific community, this is what I take issue with. Like anybody that's worth their salt, has done bench work with nucleic acid, they would know this, and they should, again, you should have a lot more support than you have. That's all I guess what I meant.
Yeah.
Like,
Oh,
but
thank
go
you
ahead.
for saying that. But yeah, that paper came out and it found DNA right on the line, a little bit under. Maybe one was like 20% over the 10 nanogram limit, like 12 nanograms or something.
And is this was a vaccine company funded project, or you
Well,
said
it's not very clear. That's the one issue with the paper is they did not disclose their BioNTech support. Their BioNTech support is through a secondary party. I can't even pronounce it. It's some institution that's funded by BioNTech, right? So other people dug that up. That there's BioNTech money in this. And there's a second preprint that's out very recently from Slovakia, from a vaccine research institution over there as well. And they don't have money from BioNTech or from Pfizer, but they do have it from some other mRNA company. I'm forgetting the name of it, but there is some vaccine bias, I'd say, in that paper. And that paper does the same thing. Is it uses phenylchloroform and ethanol precipitations to wash away all of the small material and declare there's nothing to be seen here. So the measuring the small stuff is really hard. You really can't do it with PCR because PCR kind of chokes out below 70 bases. And Oxford Nanopore, which we use, also doesn't do a good job at counting the small molecules. The part of their method is selecting for large molecules because they got such great read lengths on that platform. They want to get rid of all those small molecules, but it's a great platform for telling you what the long tail of the large molecules are, which we did in our paper and showed that some of them are 3.5 kilobases. So the DNases are in there. They're doing something. They're just not erasing it completely. And this is something that was documented by the EMA. They noticed when Pfizer gave them material that it varied, like the amount of residual DNA was varying like 200-fold in various lots. So they noted that hey, this is a problem you have to clean up, but I don't think it ever got cleaned up. And even the most recent lots that have come out, we've looked at the XBB lots and some of the more recent variants, and they're still equally contaminated as the original ones.
But okay, so there's so many questions I have for you about this subject. I mean, there's so again, it really shouldn't be that controversial. You're just analyzing nucleic acids in a tube, like there's a liquid, there's nucleic acids in there. What's the composition of the nucleic acids in this tube of liquids? Why is this work so controversial? What's the problem? You published this paper, and you're quantifying how much is in there, right? Using various methods. What's the basis for these calls to retract your paper and
So
whatnot?
I think that's because our paper demonstrates fraud, and that reverses the PrEP Act, and the liability then changes. And so as a result of that,
Your
that's
paper is your paper demonstrates fraud, or it could be used as evidence to show that fraud was committed, or is that
yes. I mean, there's the process 1 versus process 2 thing, and to be fair, Retzis Levy and Josh Gutzkow are the first people to really publish that in the BMJ. But
okay?
our work documents that there is a material difference. We can find DNA sequences that were not disclosed to the FDA that are part of their plasmid in a lot of these lots above the limit.
But aren't they immune? Aren't they immune from any?
They're not immune if there's fraud. So the PrEP Act only protects them if there isn't fraud. But if there's fraud, then it can get reversed. And then suddenly they're liable for all the vaccine injuries, which would bankrupt them. So I think that's the motivation for them to really attack the hell out of the paper the paper does discuss that even the 10 nanogram limit that the FDA has, that was contemplated off of naked DNA injections, which we know have a very short half-life in the blood due to DNases. But once you lipid nanoparticle encapsulate those things, that limit is irrelevant. And it's doubly irrelevant if you have mammalian origins of replication in there, because you can get a small amount in that can amplify. So our paper demonstrated the DNA was in fact in the LNPs. We did a very simple experiment where we just treated the vaccines with more nucleases, several of them, DNase 1 and DNase XT. And DNase XT is one from New England Biolabs that's supposed to process RNA-DNA hybrids. And when you treat the vaccines with this, you don't get a change in the CTs at all. It tells you all the DNA is not nuclease available. It's inside the LNPs. And so that demonstrates that, and that's perhaps not a surprising finding. You'd expect the DNA to be wrapped with the RNA in the LNPs, but we were just kind of curious how much was outside the LNPs versus inside, and we get almost no change in qPCR signal after we treat with these things.
But Kevin, this is one other question here. Like, these are vaccines that are still on the market. Like, are you just referring to things that came out 3-4 years ago, vials of vaccines? Or are you talking about?
The current ones still have the problem. So we've recently got
what?
XBB and they're more recent vaccines. I don't think those made it into the paper because the paper has data from whenever we did that work years ago. But David Speaker has since gone and tested other vaccines with the same methods, and the more recent ones are just as contaminated as the original ones.
Wait a second. That makes no sense to me. How is that possible if we've known this for a while? I mean, again, this isn't rocket science here. To have DNA, it makes sense that there would be DNA contamination, the way they change the manufacturing of it, the way they use DNases to chop up into little fragments, which made their lives harder to get rid of it. Knowing this for years, why would they not have just introduced an additional purification step to get rid of that?
I suspect there's a couple things going on. One, they don't have any liability, so why change? And two, they've gotten the regulators now somewhat committed to this narrative by getting them complicit in the problem, by having the regulators defend, hey, we don't think this is an issue. We're trusting the pharmaceutical manufacturers that the numbers they measured are real. They never measured them on their own. It's an independent regulator. They began publicly defending the fact that this was misinformation. This was conspiracy theory. These are anti-vaxxers.
But what's the conspiracy theory? You're just talking about molecular biology. You're doing measurements of nucleic acids. What is the conspiracy?
I think it's easy for them to say they're anti-vaxxers. We're not paying attention to this, and
sure.
They went on the
Anti-vax
record.
away, but you can do. I mean, you're a researcher. Whether your opinion on vaccine, they could. I mean, there's a big push for reproducibility now.
Yeah. And ironically, it was reproduced in an FDA laboratory, and they still haven't changed their tune. So the statements they've come out with is that, yes, it's there. We agree Pfizer didn't disclose this and should have, but we don't think it matters because it's too low in quantity and the DNA is not functional. And that last piece, I think, is where they've missed the boat. I mean, yes, it's too high. We know it's too high. They'll eventually figure that out once they realize the PCR scam that Pfizer's pulling on them by using one tiny amplicon in an irrelevant region of the plasmid. But if they use more than one amplicon like we do for COVID, they'll soon realize that there's a reason Pfizer chose that amplicon, and they didn't use the spike amplicon that they have documented in the EMA. So in the EMA documents, they do have a spike amplicon they use to prove the spike protein or sequence is in the plasmid. They refused to use that amplicon to measure the DNA concentration. They went and made another amplicon in the part of the plasmid that gets digested very readily. And then refused to use fluorometry either. So even though, because I suspect when they use that number, they don't like the number they get. But when they go and say it's not functional, it's like you cannot actually manufacture a plasmid without the promoter of the antibiotic resistance gene. So it is functional for manufacturing without a doubt. And secondly, that piece of DNA, there's all types of literature on SV40 promoters being nuclear targeting sequences. They bind P53. We also know that there's a paper from Nickels et al. That shows that there's somatic hypermutability elements that basically attract ApoBec and trigger a whole ApoBec pathway. So there's functionality there. The mammalian origin of replication is definitely functional, and that is something that can make more of itself once it gets into a cell. So they've tried to claim there's no functionality here, when their own documents declare that the Pfizer is obligated to annotate every single open reading frame, even if they don't know what they do. And they didn't do that. So there's a lot of contradiction in their regulatory documents and what they're telling the public. And so I think they got themselves stuck in this narrative of having come out saying these few lies on behalf of their sponsor, and now they can't back off of it.
Well, so I guess here's the problem I guess I have. I can see why you're a target for people coming after you, because you could stick with the molecular biology and the analysis, and all of it says that there's something wrong and something needs to be fixed, and then they didn't fix it, oddly, with the current manufacturing. But then to take it a step further and impugn motives or whatever, that may be where you get. But if you're saying, I don't know, I guess frankly, I'm appalled that they're still manufacturing it in a way that has the contamination, and then they're claiming we don't think it matters. That's ridiculous. That seems to me to be completely irresponsible and
So there
reckless.
are some very good reporters in Canada that have been doing ATIPs, the equivalent of a FOIA here in the United
Right.
States, against the regulators at Health Canada. And while Health Canada is upholding the same narrative that the FDA is, that these are too small to matter, it doesn't, they're not functional. They're privately, they got their emails, and their emails to Pfizer are telling them to get rid of it. So
Right. Okay.
what they're
Yeah.
telling the public is different than what they're telling Pfizer. And
Kevin, that's so disgusting. I see. So how are you not just angry every day? Like, I don't get how you're so calm. Oh, I know. It's the marijuana, right?
yeah, there
Is
you
that
go.
what
It is. Yeah. No,
Okay.
yeah. If you talk to Alex Berenson, it'll make me violent. So just wait.
Oh, that's right. I forgot. No, Kevin, I'm just astonished that like this isn't well, you know what? I'm not surprised by anything that's happened in the sort of
Yeah,
post-COVID
I mean,
world.
the other detail that came out of those reporters, Scoops Magoo is a good person to follow. I think I'm forgetting her Tamara Ugolini is another one, and Sonia Elijah. Those three reporters really did a good job tipping these things. And one thing they found out was that there is this limit in the FDA language that it should be less than 200 bases for these naked DNA injections, because those degrade faster, and the smaller it is, it's unlikely to be functional. But they're on record asking Pfizer, well, what's the length of these things? And Pfizer came back saying, we don't know. And we're going to have to go build an assay to figure out the length of the DNA. And then they turned around and told the public it's too small to matter.
Maybe, you know what I think it is? I think the money that they could have spent on the R&D was spent on the commercials. I don't think they
Absolutely.
have a
Yeah, right.
progress
Buying,
there.
getting Jimmy Kimmel and I
Well,
guess Bill Burr
Travis
to dance
Kelce
around with
image.
vaccine vials.
Right. No, I'm sorry. I had to make it. Since I may not walk this planet much longer, since I'm interviewing you about this, I'll
Yeah,
no.
right.
No, can you talk a bit about like the threats that you faced, or like whether it's legal actions or harassment and all kinds of like what has happened to you because you dipped your toe into this
Well,
space?
I've had my account on Twitter got canceled a couple times. When bringing this up, I had my home address go for sale on Twitter Spaces by a bunch of people who don't like me. That was fun. I've had people send me some legal letters. Our website got hacked. I've had my account on LinkedIn impersonated with some other person who's spreading profanity throughout LinkedIn. So there is and the interesting thing about our website, when our website got hacked, somebody hacked it and put up a Cialis ad so that our website would go to a Pfizer product. Uh, uh, yeah, so I don't have proof that they did it, but it was kind of ironic that they put up a Cialis ad on our website. But I don't know who's behind all these things, but there's certainly we do know the Biden administration had a lot of funding going out to diminish vaccine hesitancy, and so they were funding these groups like Project Halo and Shots Heard, I think, is another group. They go and antagonize people like me online and try to get our licenses removed. Now I know a lot of physicians that got hit by that. They have licenses that they can be targeted. I don't have a license they can take, so instead they try to drag me through the mud in a variety of other ways.
And so have you spoken with members of the United States Congress about like Ron Johnson, right?
Yes, yeah. So Ron has been on top of this from very early on. Actually, he saw the preprint, and he also entered the actual published paper into the congressional record very recently.
I saw that, yeah.
So he's very attentive to it. He's aware of everything going on at Pub here as well, and I call it Pub Smear.
Yeah,
But
what can you get into the can you get under the PubPeer thing before you say like we return to
so
Ron?
there's an organization called PubPeer that is funded by the Arnold Foundation. The Arnold Foundation got their money through some Enron tycoons, and they also fund Retraction Watch. So you'll see that whenever PubPeer, jumps on a paper, Retraction Watch is soon to follow, echoing the same complaints. So these groups go and target a very selective group of papers they don't like. They went after Wafik Eldiari. He's in the ASIP committee now, but Wafik, there's a gene in the human genome named after him called WAF1. He's one of the people who really charted out the P53 and P21 pathway. 100,000 citations, director of the Cancer Center at Brown. And when they went after him, they didn't go after. So he published one recent paper demonstrating that the spike protein in the virus downregulates P53, and made a one-line comment in that paper that this is very similar amino acid sequence to the vaccine. And that was the line he crossed. And they went after his papers from 20 years ago, where he doesn't have the Polaroids of the gel images anymore, or the people who worked on those things are now long gone. Some have passed, some are in different labs. So they're going after the papers he can't defend because they're like 20 years old, and just trying to wreck his reputation, because he was at one point being considered for the head of the NCI, and they wanted to have no part of that. So they went and have just completely hammered and harassed him at Brown. So he's gotten a taste of it. Sabine Hazan has had. She even had a paper, a hypothesis paper on ivermectin and bifidobacteria. It wasn't no data, just a hypothesis, and they went and retracted that. So they have an extreme bias in what.
But wait a second. This is confusing to me. When you say they went and retracted it, you're saying that these organizations have the power to put pressure on journals in order to get the journals to. How does that work?
So they have. There's a handful of people there, but they all have multiple accounts, so that it appears as if a paper has 30 or 40 different people attacking it, when it's really one person with multiple accounts that are making lists of complaints. And the complaints aren't always real. Like we had a complaint recently thrown up from one of the accounts there called Cheshire, or Kevin. He's admitted to his name is Kevin Patrick. But he went and claimed that we cited a paper, the Klinman paper, which demonstrated that, or just spoke about how small DNA can still integrate, like down to 7 bases or something. And so he fabricated some language out of that paper and submitted it to autoimmunity, saying these guys are misusing this citation. It doesn't say what they claim it says. And someone else on Twitter went and double checked all their work and was like, "No, you made that sentence up out of thin air. It's a completely fabricated complaint that you sent into autoimmunity." So there isn't any. There's asymmetry here.
Wait a second. He didn't fabricate it. I believe I saw he said AI fabricated.
Oh yes, he blamed it on ChatGPT, which is hilarious because those guys
makes sense, frankly.
They accuse everyone of using ChatGPT to write their papers, and then they go ahead and use it to falsify stuff against it. You can't make it up. So that was an interesting excuse, but there's also been cases like earlier this year in January. One of the members of PubPeer got angry with me about some debate we're having online, and said, and published a tweet saying, "I look forward to retracting all your papers in 2025." He didn't pick out a paper that had a problem. It was like, no, I'm just targeting you. And that's a violation of COPE guidelines. I sent that to his university, who claimed they were going to discipline him for it. I don't think it ever happened.
Nah, no one gives a crap.
So I know I'm targeted. They want to go after my papers. They've announced that in advance.
But wait a second. What's the basis for retracting your paper? You're just like measuring things, like you report the results, and then is it the discussion section? Is it something?
Yeah, no, they haven't. I haven't seen anything on PubPeer that is that wasn't already raised and traversed in peer review. Which is, I've encouraged the journal, like, just put the peer review public, because then all this noise goes away, because we already traversed all this stuff in peer review. But they went after some typos, like we misspelled the WHO, the World Health. I think we had it as a World Health Authority or something in there instead of organization.
You misspelled WHO, Kevin.
I know, it's a mortal sin, right?
Again, though, let's go back to the marijuana. I mean, we can blame the marijuana. No.
Yes, that'll be the excuse. I'll put that in my acknowledgments next time. I had too much CBD when I wrote the paper.
But this is insane. So actually, one of the issues that I'm very passionate about is how terrible the journals are for science. So one solution to this sort of the weaponization of retractions is in fact to just get rid of journals.
Yeah.
Which is what actually Jay right now has the ability to flip the switch to get rid of journals. Jay Bhattacharya, the current director of the NIH, um, solicited comments for how much of the NIH grant money should be allowed to be used to pay for publication fees. If he correctly says that that number should be $0, then a lot of the journals won't be able to stay in existence, and we will get to a post-journal world where you can just have people make comments and not use retraction. You have post-publication peer review. I don't know what your thoughts are on the journals, but
So I've gone deep into this because I was really skeptical of the journals when I got into the cannabis space because they didn't like to publish that work either. It was very controversial, and so I actually did an experiment on this where we published a preprint um, where we basically set up a crypto incentivized direct peer-to-peer review, no journal, right? Well, like, what do you need the journal for anymore? To typeset? I mean, AI can typeset, right? So they used to have printouts, and I get that that was expensive, and you needed a paper mill to do that stuff. But right now, the cost of printing something is zero on something like Zenodo or OSF or any preprint server. So really, the value is in the peer review, and none of the money goes into that process. The money all goes to the journals. They keep your copyright, and they charge people $50 for everything they want to print, and then they get subscriptions and pharmaceutical advertisement money, which corrupts the entire process. But the reviewers themselves get zero, and that often creates really bad economic dynamics. So the idea here was make an Austrian economics argument for peer review, where it's more like an Uberized market, where you have reviewers that are paid transparently, their CVs are put online, the reviews are put online. And if reviewers start cheating the system, they'll get a reputation for that and they won't be hired for the next one. And you can pay more for review if you need it done fast, and you can pay less if you don't need it done quickly. It creates a way of triaging the review process. Right now, there is no triage because it's the same price for every paper. And if it's a 100-page paper with 100 authors, you get the same process as a one-page paper with one author. That doesn't produce good outcomes because most people are going to do, the only people who really want to do review, because it's a lot of work, and if you're putting your name on, your reputation on it, are people that have a vested interest in the outcome of your work, which creates conflicts. It's usually your competitors that want to put sand in your gear, slow you down, read your paper, incorporate your ideas into their next thing, and kind of get one up on you. So we did this project where we sequenced the cannabis genome. We got it crypto funded, and we sequenced the genome and had the paper published in about summertime. We got the money in July and published it August 18th. And then we put out a bounty, $500 upfront, $500 when you're done on the review. You got to put your name public, and you got to put all the reviews public. And we'll pay you in cryptocurrency. And we had reviews back in a week, and just published it that way. So I think there's a way to do this with these immutable ledgers. The thing that's wonderful about Bitcoin is you can't edit it, and it gives you a timestamp for when you submitted it. And now you can't put the whole paper in there, but you can put a hash of the paper and a link to the paper if you know how to use Bitcoin. And you can of course facilitate international transfer of incentivization with cryptocurrencies. You don't have to worry about PayPal going to Egypt or odd countries when these crypto bounties can go anywhere. And a lot of academics hate this because they're like, you're putting money in peer review. But the reality is the money is already in peer review. You're paying $5,000 per paper. It's just none of it's going to the people that actually provide an equality. It's going to the journal, who's then turning into a pharmaceutical advertisement firm. The reviewers themselves get nothing, and I don't think you're going to get a high quality system with zero price signals on the people who are ensuring quality.
well, yeah, and I certainly have been involved. I mean, I've had conversations about, paying reviewers and what that would look like. I mean, I don't know. Frankly, a lot of papers that are on the preprint server, they don't really, I mean, we don't really need peer review for a lot of these papers. Like in
Yeah,
a
no,
sense,
some of them are in silica. You can replicate them on your computer very quickly.
yeah. And I mean, but there is, I actually think like when eLife, the journal eLife first came out, because they were HHMI funded, they had all this money and they had the ability, if they had wanted to sort of revolutionize journals by paying the reviewers, paying for reviews, but they
Yeah.
chose not to back then. I do actually like the idea of compensated peer review, and I wonder if there's a way that we can have such a thing in the absence of scientific journals altogether, like just with preprint servers and
Yeah, I mean, having been on like the ABI side of this, when we built the solid sequencer, of course we aimed to try and get a paper out. But what was more important to the company was to actually get other people to use the platform and replicate and show us our mistakes, right? And in those cases, we were dropping easy 20, 30 grand into a laboratory to get them set up and show us the defects, right? Now, one-page ads in Science back then were about the same price. So it had in my head, I was like, we should be setting up peer review in a way that you could actually afford someone to replicate it if you chose to. Like you'll put out a $20,000 bounty to actually go buy the equipment and repeat and publish what you find. And yeah, that might take longer. Maybe that's a platinum level review as opposed to just an in silico check on your data. But there isn't really any segmentation in the quality of review, right? It's like it's peer reviewed or it's not. It's binary. It's not like has it been reproduced 10 or 20 times, and has it actually hit the bench? Did someone replicate this on the bench, or did they just read the paper on a weekend and decide, oh shit, my time's up. I got to get this review back next week, and lob something back over the fence somewhat hastily. I think when it's anonymous peer review, it's easier for it to be hastily done because people can do that. They can be like, the shot clock ran out. I got to get the review back in. It's done pretty quickly. When your name gets put on it, things change. And I think we could build an entire credit system that would highlight really good reviewers. They would go for higher rates. They would capture, companies would go to them as the people to reproduce their work because they know they've got great scores and they're trustworthy. There's a lot of money, I think, on the industry side that would be willing to pay for a higher level of reproduction than what we get just through current peer review. Because I know I've been there and we have forked out millions of dollars at ABI to get people to kick the tires on solid. And that didn't go to the journals. That went directly to labs that we collaborated with. Most of them eventually wrote a paper, but it's something that I think when it comes to reproduction, if you want to solve the reproduction process, people have to actually get paid to reproduce it.
right. And yeah, and market incentives, I guess, for
Yeah.
you're the business person. Can you go back real quick? Because I cut you off. You were talking about Ron Johnson. Is Ron Johnson going to be leading the charge to sort of get safer, whatever, like, or at least answer, like just manufacturing procedures that are better for the American public? I mean, it seems odd, like that this isn't.
So I know what you know in terms of what I've seen him do publicly. I don't want to put any words in his mouth outside of that, but his what I saw at his Senate hearing, he's going after corruption in science, and so I know he's attuned to this retraction bias that's going on. That there are weaponized groups out there, and we don't know how they're funded. They don't have to disclose their conflicts in taking down a paper, where the authors have to, right? And there's a tremendous amount of work on the author side of getting a paper through peer review, and there's almost very little work setting up a civil attack, making it appear as if there's hundreds of people who don't like a particular paper, so the journals get scared and pull the plug on it. The challenge, I think, and the frustration people have as authors is that you have to pay them like, $3 to $5 grand to get your paper peer reviewed. And it costs very little for a mob to go and attack it, take it down, and your money's gone. Right. The journal doesn't give you a refund.
Well, and then it's a black mark on your career. Then it makes it harder for you to get grants. I mean, and here's, while you're talking, I'm also thinking about the difficulty I've had, like talking to my colleagues about this issue. And I think the big problem is there's only a small sliver of people where this matters. Because most scientists, they don't have any problem. They don't encounter any problems with this because they're not doing anything that's at the edge of controversy.
Right.
And so people like yourself and others that are doing really important work and then get attacked by these mobs, the broader academic community just doesn't really want to pay attention because you're a small subset of most of it's working fine, right?
I think there's obviously some centralized funding bias there, right? All of those academics are getting funded to look in a certain direction, and so they're going to publish in that same direction, and they're not going to run into this.
Right. Well, that's the thing, but they can't. Sometimes they do, and then they're like, "Oh crap! I wish we would have fixed this." It's frustrating to me to not be able to get scientists to care about this because this should be on the forefront of every scientist's mind that we are providing, truth and reproducible. We're providing reproducible, trustworthy data to the public. but when you hear a story like yourself where there's mobilization of these collectives that just try to get a paper retracted, and then you get branded as like a non-credible scientist, it's that to me is sort of that really
It's
speaks
very
to how you.
dangerous because it's very consensus forming, and science really isn't about really driving consensus. It's about challenging them, and so that is going to that I think that's what leads us into SSRIs, into statins, into Tylenol, into all of these stories we hear where science was settled for 10 years until it wasn't, right? And that is the same thing happens in the cannabinoid space, right? So it is it's really a dangerous addiction that scientists have, which is to try to continue to reproduce the narrative that's already out there, and anyone who tries to challenge it gets thrown off the boat. You find I mean, I think this is what got us into COVID, right? You find an entire society going insane because the funding is coming from one point source, and people who step outside of that become fringe, and the fringe people get labeled with retractions and tossed aside. So you can have a huge societal lemming effect like this, where everyone just runs off the same cliff at the same time because they don't have an environment that affords people with opposing views to actually nurture.
So the last thing I wanted to talk to you about, which you brought up before I started recording, is you had mentioned, I'm very passionate about gain of function research and the dangers of, souping up viruses to make them more transmissible and more infectious. You drew my attention to I guess some work. Well, there was a paper that you flagged for me. Oh, here it is. It's called, it was published in 2022, October 2022, and it's called Persistent Non-Viral Plasmid Vector in Nasal Tissues Causes False Positive SARS-CoV-2 Diagnostic Nucleic Acid Tests. Do you want to explain to me why you thought this was related to stuff that I was involved in terms of regulation of gain of function research? Why is this paper how did this pique your interest?
So this piqued my interest because I've been looking for Pfizer DNA in a variety of places right now, that not just in the vials. There's a couple of studies in the SRA, the short read archive, where people are doing RNA sequencing between vaccinated and unvaccinated, and we pull down their data and we can find the plasmid sequence in those patients. Don't know if it's integrated; it's just there, right? So and I've been paying close attention to that because in our own efforts, we've been trying to sequence some biopsies, and occasionally we'll get hits that look like the SV40, and we're like, okay, is that really the vaccine, or could it be another plasmid from someone else's laboratory who handled this? And that's what got me down this rabbit hole, is that there are other plasmids, research plasmids out there that share sequences to Pfizer, because Pfizer used a very commercial vector known as pcDNA 3.1 is the backbone of their plasmid, close to it. It's probably the closest I could find in Addgene, which is a database that holds a lot of these plasmids. And so this paper popped up on my radar because this was a case where a laboratory was working on a SARS vaccine, and they cloned in nucleocapsid instead of spike into the plasmid that's very similar to Pfizer's. And this thing spread all over their laboratory, infected the lab staff, and got outside the lab to a housemate. Now, if that plasmid were making, I don't know if nucleocapsid is particularly, pathogenic like spike, but if it were making spike, and I know there's lots of labs that are studying vaccines with spike in plasmids just like this, these things can replicate in E. coli and in mammalian cells, and so they can spread amongst the lab staff and get outside of the laboratory. So that to me is a gain of function hazard, because if there is such a thing as spikopathy, the patients that are accidentally spreading this, or the lab staff that are getting it outside of the laboratory, they're going to create either false pandemics because the PCR tests are getting triggered by this, right? Um, uh, and potentially illness in people outside the laboratory, and they're never going to suspect it to be the full virus, or they may not know where it's coming from. So I think those plasmids that people are using to make vaccines, they need to be careful to make sure they're only in bacterial hosts. When you put them in a plasmid that can jump between bacteria and mammals, they're effectively zoonotic, and you're encouraging something to hop between E. coli and mammalian hosts. So it's an area that I don't think the gain of function debates actually looked at, is what is leaking out of all the laboratories that are trying to make vaccines. The plasmids that they're using have been documented to leak in Seattle. Are those leaking in 100 other laboratories right now? And what is that doing to our PCR surveillance? What is that doing to people's exposure to the peptides that those things are making? If they're pathogenic peptides like spike, that's another avenue for disease that's going to probably go under the radar and be misattributed to either the virus or to the vaccine.
So, okay, so this is something, and I appreciate you flagging this for me. So I'll take a look at this and see if there's something related to, yeah. There's ongoing efforts to regulate dangerous gain of function, and depending upon what policy emerges, this is definitely something I'll keep an eye out on for whether or not these policies cover stuff like this. I do want to ask you, do you ever do anything that's non-controversial? Are you always fighting with pharma companies and like,
Yeah.
I'm concerned. Like every single thing you're doing it seems like it sort of puts a target on your back, Kevin.
I I I I I don't know where I went off the rails on this, but I think I got some frustration in me when we built the solid sequencer and we could suddenly sequence genomes for like $3,000, and it seemed like a logical thing to do with tumors, like sequence a tumor, sequence the patient, look for the mutations that differ between them, and see if there's a druggable target for these things. And then it just got stalled in like FDA morass and red tape of people actually being able to deploy that. You probably remember when 23andMe more or less got neutered, not recently, but like 10 years ago. The government came in saying this direct to consumer stuff is dangerous and you can't do it anymore. You have to go back to just doing ancestry. But they had these great reports that would read your genome and tell you if you had ApoE variants or BRCA1 variants. And so I thought the genomics field was going to go in that sort of personalized, direct to consumer direction, and I think that got shut down by the pharma industry, and the FDA just put, and maybe even part of the medical establishment didn't want that type of direct to consumer information out there. Um, and that's when I started getting interested in other medicinal organisms that people could theoretically grow in their backyard and escape this whole paradigm. And that's what drew me to sequencing cannabis plants. We've done Psilocybe cubensis as well, which is a magic mushroom, which has got a compound now that's in fast track at the FDA. It's called psilocybin. That looks like it's phenomenal for depression. A lot of these compounds that are illegal are actually safer than Tylenol, and they're outlawed, and no one, it makes it very difficult for people to study them. So I've been focusing on those areas because I think we can lift all boats if we can get more grounded science around the how those compounds are expressed in those organisms, how we can make them better, faster, cheaper, and how we can normalize them a bit more. The therapeutic index on a lot of the SSRIs out there are awful. It's like 10 to 20. You can barely measure the therapeutic index on, or at least the LD50 on psilocybin or CBD or THC, right? But these compounds are illegal. And for those not familiar with the therapeutic index is the ratio of the LD50 to the ED50, right? Lethal dose 50 to the effective dose 50. You want that to be as wide as possible and to have a very low dependency scale. And you look at the drugs we use in psychiatric medicine, like lithium, and it's a 3. Like if you're off by 3 with lithium, you could kill half the people, right? Whereas the compounds like psilocybin, they can barely measure it. It's thousands, the ratio between effective and lethal dose. So we have a tendency in the country to criminalize drugs that threaten pharma, safe drugs that threaten pharma, and I'm doing what I can to try to reverse that by bringing more sequence information and genomics to the fields that where these compounds are being neglected.
I see. So that explains. I did want to know. I guess one question I had that emerges from what we were talking about with your work on the mRNA vaccines is, recently I think announced the NIH would be sort of deprioritizing using mRNA or funding mRNA. Actually, what maybe my question is, do you see potential in the mRNA platform for
Oh, yeah.
treatments and therapeutic?
Yes. I mean, it's really disease dependent. I mean, there are cases where you have a terminal disease and the risks for using these are maybe lower than the disease risk, right? I mean, Onpattro is one where they're using microRNAs. And of course, those are chemically synthesized. They're very pure. They're infused over hours. They're given with immunosuppressants in advance. So it's a very different game. There's other cases where they've used gene therapy plasmids. I think Duncan et al. Has a good paper in New England Journal of Medicine where they use a lentivirus, vector, and that thing integrates about 1,000 times per patient. You think that'd be a nightmare, but those patients have a lifespan of like 10 to 20 years. So they're willing to take 10% of the patients got cancer in that paper, but they're blood cancer. But they're willing to take that risk because the disease is so frightening. So there's the risk reward is where this has got all, I think, skewed here. Is they amplified the risk of the virus. Jay knows that from the beginning from his antibody studies, and then they downplayed the risk of the solution, and then mandated it, created liability shields. So I don't believe the government needs to be funding any of this, given all of they should get rid of the liability shields. Then the pharma companies probably won't want to develop them because the risk reward will be on them, and they don't want that. But I don't think there's any reason the government should be putting in money on top of this unless they're looking for royalties. I mean, that's something that does go into the NIH, is over $1 billion of royalties for the mRNA vaccines flow into the NIH, which probably explains why they never did any work on ivermectin or hydroxychloroquine, these cheap drugs that don't line their pockets. So, I don't believe the taxpayer should be funding the development of these when pharma made $100 billion off of them liability free. Like, they can afford to do their own research.
But you do think that there are applications, that you're not completely against the platform.
No. I think that the economic incentives are all wrong. They're mandates and they're liability free, and so in that scenario, you're never going to get the risk reward right. When you remove that stuff, you can start thinking a lot more actuarially at this, being like, okay, here's a disease where you have a lifespan, you may die at age 20, and an LNP is the only solution to fix this. Fine. Yeah, I mean, you'll take the risk, but many of the people in the Duncan paper, you could see that 10% of them get lymphoma. You can treat lymphoma and live longer than 20 years. So that's a trade-off that makes sense. But no one can make those trade-offs in an environment where the risks are hidden, the liability protections in place. You can't do the calculus.
Okay, well I just wanted to make sure I recorded you saying something positive about the pharma company.
I'm not anti-biotech or anti-pharma.
Well,
It's more the economics of this that get me. I mean, I still pipette at the bench as we speak today. I run these sequencers when we have to. It's a really powerful field. You can do a lot of good with this field. I hope people don't get turned off that all biotech is out to kill them. It's just I think when it gets centralized, we run into problems.
they're just trying to make money. We're just trying to make a buck. That's it.
That's it.
Okay, this is like actually literally the last question I have for you. What's the deal with the owl on your Twitter profile? What is up with that?
That's actually my cat. That's a Maine Coon.
Oh, I'm sorry. It's not an owl. It looks like
No,
okay.
you should call it an owl because people get flagged for saying the word coon on Twitter. I've had people get blocked if they talk about Maine Coons. So
Okay, so that is your cat. Okay,
yes,
I think I knew that before, but anyway, that was one thing I've always been it's a striking image. So that's why it's always like something. I mean, I use a fox as my profile photo, so But anyway, I appreciate you taking so much time to chat with me. I really had a great time because I kind of like sort of wonking out, if that's the word, or getting a little into the science. And I hope that we can talk again about this stuff because it's actually very important and also quite fascinating.
Absolutely. I love talking to people that have experience in the genetics and genomics field because it's I find myself in a lot of these casts where the people aren't versed in this, so it's harder to discuss. And so it's refreshing to talk to someone who's got your background in this and understands the jargon.
Yeah, I appreciate that. I think we should do more of these discussions because there's very few scientists that are willing to actually have public discussions about this stuff, which is sad, I guess. But hopefully, maybe, I don't know. Hopefully we'll do another one of these interviews or maybe a Twitter space someday. Do you ever do Twitter spaces? Or?
Sure, yeah. I jump on those on occasion.
Okay, I'll find a day and time that works for you. But anyway, thanks again, Kevin. I really appreciate it. Until next time, this has been Bryce Nickels for Science from the Fringe.